PBC stands for Primary Biliary Cholangitis. It is a liver disease where the cells lining the bile ducts are seen as foreign, under attack by the immune system. Doctors can treat it with drugs, but there is no cure at the moment.
I've seen people scratch themselves so that they've drawn blood, and for those friends of mine in the patient support group, I've got such sympathy for them because they'll scratch, there'll be scarring, they'll be scratching over the scarring. There'll be other people that are so fatigued that they can hardly even walk from the car to their office or the shopping centre or something like that. I'd love the management of the symptoms to be addressed by research.
It seems that the immune system attacks a protein present in the part of the cell that uses oxygen to generate most of our energy. The cause of PBC is unknown, but over the years it has been linked to environmental triggers such as man-made chemicals.
This project was started at the request of a community group that I worked with in the North East. The group lived close to an active landfill and were interested in the potential environmental triggers of the disease.
What we decided to do was to go out into the environment and actually see whether the environmental component could be sampled and tested. Not just by traditional means, using sensitive analytical equipment to identify the classic well-known toxic chemicals, but also by chemicals' effects on cells which are cultured in the laboratory.
We worked together with a community group to identify sites where we could collect particulates in the air and surface soil in the first instance. So we sampled soils at various places around the landfill site and from other areas so we could make comparisons of the chemical profiles. We extracted the chemicals using different types of methods. We tested the soil samples for the presence of well-known pesticides, persistent pollutants and metals that are often thought of as contaminants.
But we also tested for toxicity in cultured cells. We used, for example, a special type of liver stem cell because we thought that if this cell was sensitive, it could explain what we see in the PBC disease. What we found was that the landfill soil was no worse than any other soils in terms of well-known toxic chemicals. But what we did find was that there was something toxic to our stem cells in particular in a couple of soils we had taken from around the landfill site. Interestingly, the toxic extracts almost immediately stop the part of the cell that generates most of its energy, preventing the cells from using up oxygen that they need to survive. Up until this point, we had no idea what the chemical in the soil was that was causing this effect. So we had to take a new approach. The hard work began where we tried to purify more of the toxic chemical or chemicals away from everything else using its toxic effect as its identity.
Once we thought we had enough chemical, we examined it in various ways. For example, using mass spectrometry to smash the molecule up into different fragments. By comparing the fragment patterns of our chemical with already known mass spectrometry libraries, we started to get information about the chemical structure. Doing this, we narrowed the chemical down to about 5,000 possible structures.
So we still felt we had a long way to go to identify the chemical, and called in the help of the chemists who are experienced in chemical design and identification. We finally worked out what it was when we used something called nuclear magnetic resonance spectroscopy. This uses very high magnetic fields to help work out the chemical structures. Doing this, our chemists helped us work out that the toxic chemical was an ionic liquid called C8-MIM or M8OI. Ionic liquids are being developed as alternatives to normal solvents because they do not evaporate so easily, for example. We then took the pure M8OI chemical, we showed that it had the same effect on cells as the soil extracts. What was curious was that the structure of M8OI was a little bit like lipoic acid. Lipoic acid is made by our bodies. In PBC patients, their immune system mistakenly attacks a protein where lipoic acid is needed for it to work. What we needed to know was whether the human liver could change the M8OI to a more lipoic acid type molecule. In other words, to make it a carboxylic acid. As part of the transplant research program, surgeons at the Freeman Hospital are looking at ways to improve organs not good enough for transplant. After this research, the organ is normally discarded. Using some of this liver, we isolated the main cells of the liver and we put them in culture. We then added the M8OI to see what happened. In cells from several donors, there was a rapid change of M8OI to the carboxylic acid. We then synthesized the M8OI carboxylic acid and showed that it could replace the normal lipoic acid in the protein associated with the PBC disease. So, either M8OI or any number of chemically similar man-made chemicals, as there are many, could, at least in theory, be a trigger for PBC. Whether they have caused PBC in any patients is another question to be examined. But this research, which has pulled on the expertise of a wide array of scientists at Newcastle, and in fact working with colleagues from Sweden and Egypt, has shown that this approach can make a real inroad into working out what might cause a disease. And of course, this approach could be applied to a range of concerns about the effect the environment has on human disease.
So what's the future of our research? At the moment, we need to secure funding to extend our study. We aim to investigate how these chemicals act and interact when they're in the environment, find out how long they remain in the environment, and how people could possibly be exposed to these chemicals in their day-to-day lives over time. For example, through particulates in the air, through contact with soil or water. Then, having scientific evidence about exposure, we can start to answer questions like, how do we as a society measure the impacts of new and novel chemicals on the environment and human health? Where should we be routinely monitoring these chemicals, for example in waste management or disposal practices? And what measures do we need to put in place to ensure our future environmental and human health?